So I just want to show you where your primary studying of images should be, okay? So for the brain and spinal cord structures, what we want to look at is going to be from that McGraw-Hill website. So let me get out of this. So if we go into that cadaver dissection tool that we've been using, now we're going to go into the nervous system model. And so you can see that we have a spinal cord topic, peripheral nerves, brain, cranial nerves, sympathetic, parasympathetic, taste, smell, hearing, balance, and vision. So we'll hit these sensory ones next week. For now, we're going to focus on spinal cord, brain, and cranial nerves. So let me just start with that because it's the easiest. So with cranial nerves, we look at the inferior brain, and the cranial nerves are all the nerves that emerge from basically from the brain stem. And like I said, they control the face. And so this is, and yes, the list is now on Canvas. Hold on, let me show you where I put it. So if you go back into that online lab resources, I put it at the very top, so you can just download it right there. So learning the cranial nerves is a traditional part of anatomy and physiology. And so luckily I remembered that that's something we should do. And this is going to be your testing image for cranial nerves. It's also going to be one of your brain ones because there's a lot of brain structures on here too. So if you look at these guys, we're going to talk more about cranial nerves later, but basically there are 12 pairs. They come off of the brainstem, and they start with the olfactory bulbs, which receive smell information, and the optic nerves, which receive visual information, so from your nose and from your eyes. And then we have other pairs coming off down here, all the way down to, where'd you go? These are in alphabetical order, which is not what I'm used to. These guys, which are the hypoglossal nerves, which control your ability to move your tongue. So all of these little guys sticking out are different cranial nerves. And so you just want to be able to identify them for lab, know their names and their numbers. They are numbered in Roman numerals 1 through 12, which is why that's how this is written on here. And that's how I put it on the list too, so you can find them both by name and by number, although they have them listed in alphabetical order, which is weird. So that's that. And of course, things that I didn't really show you guys before, but hopefully you found out. So if you go to the information button after you've selected a structure, it'll give you information about it. You don't need to know this detail of information. You kind of just need to know the general stuff, and that'll just be for lecture. So it'll be in my lecture slides what you actually need to know about this. So there's a lot more here than you need, but it'll also tell you how to pronounce them. Glossopharyngeal nerve, which is cool. And you can also pick that just in case there were other views. There aren't there for this one, but you know, if there were, and then you click on the clipboard to take you back to the general list. There's other things you can do in here that you can play with, including take pictures for studying offline. If we look at the brain, this is going to be your most complicated part. So there's 1, 2, 3, 4 views, and then a close-up of the inferior. So the inferior close-up, you really don't need per se, because mostly what it's showing you is the blood supply. This is that blood supply that I showed you before, and it's not on your list of things you need to know. But some of these might be useful too. So for example, the mammillary body is right there, and that's going to be one of the structures that you learn. Nothing else is super interesting on here, but look, here's the pons. So that's the kind of stuff we're going to see on the inferior view, or things like the pons, the medulla. Here's the cerebellum. There we go. Stuff like that. Apparently, you can pick brain, in case you're wondering where the brain is on here, which is everything but the beginning of the spinal cord and the cranial nerves. Your coronal section is viewing this from the front, which is kind of interesting. Your lateral is actually in layers. So their first layer is skin. Your second layer would be bone, but they cut that away. So this allows you to see the dura, which is not actually on your list. But this is going to be more useful. So this is a lateral view of the brain with the dura cut away. Your cerebellum's down here, and different brain regions, like your central sulcus is dividing up your frontal lobe from your parietal lobe. And then if you take it here, this is a mid-sagittal section showing you the falx cerebri that separates the right and left hemispheres, for example. This is your corpus callosum right here. And you can see this is midbrain, pons, medulla, and you can see that the medulla transitions to the spinal cord as it exits the foramen magnum. And your cerebellum sits right here, right above the foramen magnum. Ooh, and here's your pituitary gland right there. And then if you go here, then this is the mid-sagittal section. And so this is a good one to study because our brain models that we would have studied can split in half like this. So this is a good view to see because it's going to correspond to model views that you may encounter later. And also because there's a lot of structures you can see on here. And then we've got a superior view, which again has the same layering. So you can see dura, see without the dura. You can see it as a sagittal. So we would have gotten to slice up the sheep brains like this too. So this is a great view because you can see these are the lateral ventricles. Lateral ventricles right here. And there's, please tell me that it's that. Nope, hold on. Nope, it does not have choroid plexus in here. That's okay. There's a lot more on here than you need, so please go with the list and don't worry about things that are not on the list. But it even shows you things that I should have put on the list, but that's okay. Here's the insular lobe right here. Remember the insula? You don't actually need to know it because I didn't put it on the list, because in models you can't usually identify it very well. And then this layer is actually the skull without the brain, which is kind of cool. You don't need that view though. And then what's the other one? Did I show you everything? Those were superior, lateral, coronal, inferior. So yeah, that's pretty much, those are all your brain views. Your spinal cord has the cross-sectional view. So if you go to the typical spinal nerve, that's your cross-sectional view. So this is usually the most common model that we have in lab and a good way to study it. Now we usually have just this. So this is much more proportional than usual, but it's still just as good and is going to show you all the things that you really need to know. And it's cool because you can see how the vertebra sits compared to the spinal cord, and you can see the dura and the epidural space and the subarachnoid space where the CSF is and all of that stuff, which is pretty cool. If you look at the overview, it shows you the entire thing, and it shows you what it looks like sitting in there. This is with the dura on it. This is with the dura removed. And then this one is like with everything removed. So now you're really just seeing a naked spinal cord. So you can see the cervical enlargement up here, And you can see the lumbosacral enlargement here, lumbar enlargement, see? Conus medullaris, coccygeal region here, filum terminale. This, as I said, does anchor it down here to the base to keep it all in place. So there's a lot of things you can see. Not all of it, again, is on the list, but some of it is. And then you can take a look at these guys like that. So specific layers, for the most part, I'm going to test you on the layers that actually let you see the things that are on the list, if that makes sense. And so for this stuff, it's again just whatever to identify the things that are on the list. And so like, for example, dorsal root ganglion, which we're going to talk about later, is one of the things you want to be able to identify. And it's going to look pretty much the same on anything, on any of the different regions. So like here's thoracic region. That's going to look like that there. So just like with the muscles where, you know, sometimes there's things deep to other things, I might have to use more than one, but it's going to be whichever ones let me actually show you and ask you about the things that you need to be able to know from the list. So for example, like the brain one, a lot of it is going to be on these lateral layers. So this one showing you the whole brain, and then this one showing you half the brain are your actual useful brain layers, because the rest of them you can't see all the brain structures because they're covered by other things. So unless I want to ask you about the falx cerebri, which I can't remember if it's on here or not. Nope, I did not put any of the Dura stuff on here. Maybe I'll add it next week because we do have really lovely images of it. That would really be the only thing I would ask you about on this layer though, if that makes sense. So if you're trying to figure out the layers, then use your list and click on things that are on the list here, and it's going to take you to the layer that lets you see that, if that makes sense. So that will tell you which layers I'll be testing you on, because they're the layers where you can see the structures, if that makes sense. So yeah, this, like for example, on the lateral, this layer 6 and 4 are the useful layers. And then if you look at the coronal section, this is the overview one is probably your most useful. And then when these are layered, it's literally slicing different places. So if I showed you this, it would be because I wanted to ask you about something like the ventricles. So like here's the lateral ventricles, here's the third ventricle, here's the corpus callosum up here. And if you go, whoa, that's too far. Like that's just useless. What can I do with that? I can't really do anything with that. So really it's like this one, because nothing else is useful here. The inferior is just the inferior, like there's no view of that. And then the superior one, again, like the only thing you can see here is dura. This is useful because we can look at the different lobes. So like frontal lobes, central sulcus is right here. But clearly these are not set up like that. So I'll probably be using the lateral for that. And then again, this stuff is just useful for things like the ventricles, which don't show up well on anything but some sort of cross section. So ventricles there, not going to use that because there's not actually any brain in there. So this one is your most useful sagittal, or superior, sorry, if that, does that make sense? So like I said, like kind of use your list, because your list is going to show you the layer that you need to see something. So, and then if you're not finding it on here, if you're having trouble finding it, you can do a couple of things. So you can search. Okay, so for example, the longitudinal fissure, there we go. It's being slow. Oh, I see. Okay, so like I didn't see it on there. So I looked for it, and now it's giving me the view, which is this first coronal view, and saying, well, look, there it is right there. It's literally the separation between the right and left hemispheres. Okay, so that's the other thing too, is you can search them as you go if you're not finding them. So again, it's going to give you the views that they're labeled on, and those are going to be the views that I use. Okay, let me show you the other thing. So the slides are on this website. You can all see this website, right? It switched. Yes, as I change tabs, you can see it, right? Okay, so this website, this page has really good slides, and I think they're done in a really good way. And we're not looking at all of them, okay? So there's only 1, 2, 3, 4. There's 4 specific slides, and then there's a couple of sensory nerve ending slides, and it doesn't really matter which one you look at. You're going to see the same thing. So you pick a slide that's actually on my list. So here is your, of the 3 spinal cord slides, you only need to look at this one, okay? Because this is pretty much what we would have seen in lab. So the cool thing about these slides is that you can kind of do a self-guided tour. So there's information on this side, and I lined up what we want to identify with what's on here. Like I literally typed it in like, oh, well, this is what it's going to tell them to look at, so this makes the most sense. So first of all, for this one, we're going to want to look at the same cross-sectional anatomy that I just went over in lecture. So I do that in part because it makes you review it again, and in part because this is, again, what we usually see. And then the other thing that's really nice to see in here is the motor neurons. So these slides are really nicely set up. So I clicked on motor neuron, it's highlighted over here. It takes me to that close-up of these guys, these big old purple things. These are motor neurons. And it's showing you where it is. It's in the gray horn, or it's in the horn. Looks kind of laterally, but I think it's actually ventral, because that's where the motor neurons are. All this other stuff is glial cells and blood vessels and stuff like that. So you want to be able to identify that this is a neuron, and then you want to be able to identify the gray versus white. So see, the gray matter is here, and you can see even on this less magnified view, the neurons are huge and very easy to see. And there's a distinct look between this versus this. So this is the white matter, even though all of this is stained pink. So this is white matter. It looks lighter because a lot of these are axons and they're myelinated. And so when you look at that myelin, and you can increase and decrease the magnification. So when you look at this, all this white space, that was myelin sheath. That was oligodendrocyte wrapping itself around, and now we're seeing empty spaces. And then like this is axon running longitudinally, and all of this stuff is axons in cross section. Now you don't necessarily need to know that. What you need to know is this is white matter in the spinal cord, okay? And then the other thing here is the central canal. So the central canal is where the cerebrospinal fluid runs right down the middle, and it is lined with ependymal cells, which makes it one of my favorite structures, because the ependymal cells are really the only glial cells in the central nervous system that you can easily identify, because they're only in one place, and they're easy to see because there's a lumen. So I enjoy them. This isn't actually the best slide I've seen of them, but these big guys are the ependymal cells. So that's why I picked this particular website, because it gives you all the information you need to easily identify things. It takes you to the close-ups. And here's the other thing too. So you can hide that description or make it come back. You don't really need this, but it does give you information about the specific slide. You can change the settings, and then I swear I saw this. Well, I don't see it right now. Maybe I looked at several different websites, so it might have been the other one. But you should always be able to screenshot this to study the still images too. And so I will take, for your test, I will take images from this slide set to identify just the things on the list that I'm telling you to know, okay? So they're really nice slides. They're fun to navigate through. Let me show you the brain. And we'll probably use this same website for I haven't looked at their digestive slides yet, but we'll probably use the same one, because if they're all like this, then it's now my favorite histology website. The only one where I didn't give you specific slides is these guys, these sensory nerve endings. We're looking at two things in here. Hey, stop it. Stop it. These are the two types of nerve endings that are easiest to see in microscope slides: Meissner's and Pacinian corpuscles. They have other names too. We'll cover those when we get to them in lecture. So if you look at the descriptions, this particular one has both on there. And so if you just want to look at that slide and just look at it, that's fine. You can also look at Meissner's just on this slide, and then both of these have Pacinians only. They will look the same in close-up regardless of the slide, which is why it doesn't really matter which ones you look at. So you can find them on your own if you want to. So the Meissner's are way up here. I always get them backwards, but these guys are super easy to find because they're gigantic. This onion thing, that's the corpuscle. So and then oh yeah, sorry, these are Meissner's. I don't know why, I always say them backwards. So this is Meissner's corpuscle, this whole giant thing. It lets us feel pressure. And then the Pacinians, where'd you go? There's a bunch of them. Or is this Meissner's? I don't know. Oh yeah, this one is. I'm sorry. I should know this by now, but I always for some reason get them backwards. So these are Meissner's, this guy right here. There's a bunch of different examples. And as you click on each little blue thing, it's going to show you them. So it's these guys. This thing lets us feel light touch. Lots and lots and lots of examples all throughout the slide. And then the Pacinians are deep. And like I showed you, let's see, go back out. They're really easy to see because they are massive. Each of these oniony things is a Pacinian. There's a little slice of one there. So you can identify them from a fairly low magnification. So there's the one I showed you before. This is a very nice slide because it's hard to find them on the skin a lot of the times. Those are all the ones I can see off the top of my head. So you can just look at that one if you want to, but they also are present here. So for example, we have some of these Pacinian guys in our abdomen. That should be it. Yep. See that? Very distinctive. So if I'm testing you, I'm going to show it to you like this. It doesn't matter which slide you look at. They're going to look the same regardless. So that's why that one's like that. But the rest of them, I picked specific slides because they correspond most closely to what we would have looked at in lab. So for example, there's 3 nerve slides, but this is the one we're going to look at because this is typically what we would look at in lab. We would look at a single nerve slide with a longitudinal and a cross section, and you want to look at that and identify basic nerve anatomy, which we haven't covered yet. So you can look at it later after we've covered it, but it's going to correspond to the same anatomy we're going to cover in lecture. All right, so that's what we're going to do for histology this time because I didn't like the fact that it was just a single picture and that wasn't always very good on the McGraw-Hill website for that. So you don't have to worry about looking at that part of the cadaver dissection this time, right? You don't have to go here. You can just look at these, this stuff. And then I'll add a couple other things next week, but this is more than enough to get everybody started. And we'll make up the bulk of what we're doing for nervous system and lab. Does anybody have any questions? Stop it.
Do we have another lab report due soon?
So yeah, the future lab reports, I haven't sent an announcement on that yet, but we're gonna just cancel that because it's too much. I mean, I haven't even had a chance to get to your last ones. I'm still trying to grade everything. I don't want to do it. And I feel like I double checked and I think that we covered the writing requirements and there will still be short answer on the tests. And so all things considered, we're just gonna not do that.
Okay.
Yeah. Especially because I'm not finding really great websites to take the place of what we would have done in lab that we would have done the lab reports on. So yeah, we're just not gonna do that. The link to the histology guide. So yeah, so the histology guide stuff that you're gonna be studying off of, the link is actually on the Word document with the list of anatomy stuff to know. And here, let me show you that again. So the anatomy list to know is this guy. So the first page is all the brain, cranial nerve, and spinal cord stuff. And then the second page is histology slides. So there's two websites on here. This first one is one that I actually like for other stuff, but it turns out that it has no nervous system actual slides, but it has a good overview of nervous tissue anatomy. Like it goes through all the anatomy stuff that we've been going through and are about to go through. So it's not a bad place to hit if you want another version of some of the stuff I've been telling you. This one has the slides. And so you go to this exact web link and you will have access to all of these slides. I gave you the slide numbers as well as what they are. And then the stuff that you need to be able to identify is listed underneath. Okay. I should have bolded these. So 1, 2, 3, 4, and then I'm just calling that 5. But like I just showed you, you can look at just that one slide or you can look at all of them to see these two different structures. You just have to be able to identify these two structures on close-up for those sensory nerve ending slides. All right, any other questions about lab stuff? Everybody okay for now? Okay, so like I said, yeah, I think that's more than enough to get you guys started. And I think given the timing of everything, I'll probably just record the cranial nerve, spinal nerve part of the lecture to get us caught up later this week and post it, rather than waiting till next week to see how far we get, because that way you'll have it sooner rather than later. And I'll send an announcement and an invite out. I don't expect any of you to show up to it, but if you're free and you want to hear it live, then I'll give you that opportunity to. Okay? But no worries if you can't make it. Even less expectation than right now. Okay? All right. So per usual, I'm working on grading. Send me emails if you have any questions, and I will see you all next week. There we go. Okay. So, yeah, we can take a quick little break again. I need to like get a drink of water, and that way you can reset. But all you're gonna need to do is be able to type the answer. So basically I put it in a PowerPoint and I'll screen share it. And it's one question per PowerPoint slide. And it's gonna just cycle through them. And so as you see the slide, so that I don't get confused, I have them as words and not letters, you know? So like if it asks you to identify something and the answer is femur, you're gonna type femur, okay? So you'll just type them in, and that way like the chat, I can save it and that'll be your answer sheet.
I'd have to type them in directly on the PowerPoint?
No. You're gonna type it into the group chat.
Okay. Okay.
Yeah. So just give me 5 minutes. Let me go get a drink of water and stuff. And
Okay.
then,
And then my screen is off, my kids are up, so I don't want them to come in. You might see somebody hit somebody with a pillow, and I don't want to cut the mute off so that they don't scream while we're doing this.
Yeah, you don't, and for this, honestly, I mean, you don't need your video or your mic on if it makes it easier.
Okay.
Yeah, because that way because you just need to be able to see the screen and type it in.
Okay. Well, thank you.
All right. I'll be back in a couple minutes.
Okay.
You chewed that up. That's great.
Hello, Allison Thompson.
Antonio. shit. I need to get